Welcome back to learning how you function. What you learned in the last lecture is how, with the simple cooperation of your hypothalamus, you can affect virtually every cell in your body. You are now spectacularly schooled in autonomic function. How your sympathetic nervous system can arouse you, your parasympathetic do exactly the opposite. A theme that came through in that lecture is the potential for your hypothalamus and your hypothalamus getting emotional input from your limbic system, cognitive input from your cortex, the ability of your hypothalamus to reach out and touch virtually every outpost in your body to regulate events all over the place there. What we'll be doing today is switching over to the other way in which your hypothalamus can regulate things all over your body. This is our second realm by which your brain has this ability to touch everywhere. And what we'll be looking at here are hormones. Hormones, your endocrine system, the ability of your brain to regulate hormonal events throughout your whole body. This is the other way in which your hypothalamus which plays just as important of a role in regulating hormone release as it does in regulating your autonomic nervous system, how it is that your hypothalamus can also alter events throughout your body through this hormonal route. Okay, we start off with some definitions. What makes a hormone a hormone? And we go to our first diagram, and what we see here is one version of things that we're already deathly familiar with, neurotransmitter release. We got our neuron here, we have the next neuron in line, we've got our synapse and chemical messengers coming out, and by definition, as we've learned, if there are things that are sitting in vesicles here, they're released, they go across the synapse and affect events on the other side. We're talking neurotransmitters. Our first definition of a hormone is derived from this. Looking here in the second diagram, what we have is a neuron, a hypothalamic neuron as we will see, a neuron releasing chemical messengers, and this time, instead of traveling across the synapse, this time they wind up in the circulatory system. And by definition, if you've dumped a chemical messenger into the circulation where 14 counties away they hit some cell and change events there, this thing's a hormone. Go across the synapse, you got a neurotransmitter, get into the circulatory system, you're dealing with a hormone. What we see in the third part here is not all hormones are derived from neurons. You can have hormones coming out of any old cell in your body, a pancreatic cell, an ovarian cell, whatever. The common theme is if it gets into the bloodstream, if it gets transported a great distance, you're talking about a hormone. Okay, this begins to give us a flavor of the difference between neural communication, neurotransmitter release, and hormonal. By definition, if a neurotransmitter merely has to go 1/1000th of an inch across a synapse to have its effects, whereas hormones have to slowly percolate through your whole circulatory system, hormones work more slowly. We saw in the last lecture voluntary nervous system working in 1/10 of a second, involuntary, the autonomic nervous system, working over 4 or 5 seconds or so. Hormones take minutes. Hormones can take hours to have their effects, much slower system. Another difference is just the sheer mechanics of the communication, who you can affect. With a neurotransmitter, the only cells you're affecting are those sitting on the other end of your synapse. 10,000 cells at a maximum. With a hormone, get that messenger into the bloodstream and potentially you've got access to every cell in the body. The third big difference between neurotransmitter communication, neural communication, and hormonal communication is in the hormonal case, you can have much broader effects on those cells. Back to our first couple of lectures, you know what neurotransmitters do. They make other cells more excitable or less excitable. All they're doing is changing something on that cell membrane. With a hormone, you can do the same thing, but in addition, you can turn on genes in that cell, you can turn off genes, synthesize new enzymes, you can cause the cell to divide, you can kill a cell, you have a much broader range of effects. Hormones, you don't have the advantage of the nervous system of causing very rapid effects, but with hormones, you can regulate all sorts of aspects of cell biology. Now, the centerpiece of endocrine communication, hormonal communication, is, as I mentioned, your hypothalamus. Your hypothalamus is ground zero for control there, and we should be able to recognize by now the two different ways in which you have regulation going on there. Back to our last lecture, and we have the hypothalamus sending its autonomic projections, midbrain, hindbrain, down the spine, and all of that's familiar by now. The general scheme for endocrine control by the hypothalamus is the hypothalamus regulates events in your pituitary gland, sitting just underneath the hypothalamus, and your pituitary, in turn, regulates events in all sorts of peripheral glands, your pancreas, your testes, your ovaries, your kidneys, so on. This would be the flow, what's called the neuroendocrine flow of communication. Now, what do I mean with this abstract term, your hypothalamus regulates your pituitary? Your pituitary regulates these glands. These arrows indicate hormones. Your hypothalamus releases hormones which affect pituitary function, in turn triggering the pituitary release of hormones, in turn triggering these peripheral responses. All of these arrows are hormones. What you see in this cascade is this flow of communication, hypothalamic hormones, pituitary hormones, the hormones of these peripheral glands. This is the flow of information. And this is still, a relatively new story in biology, and one of the more interesting ones in recent years, this primacy, this recognition that your brain is an endocrine organ. Your brain is a gland that can release hormones, and this is not what people used to think. A little bit of the history of the subject. Back at the turn of the century, there was this notion that these peripheral glands, people at that time recognized peripheral glands made these hormones, these messengers that got into the bloodstream, and the sense then was peripheral glands were autonomous. What do I mean by autonomous? They knew what they were doing. Your pancreas could sit there and figure out things all on its own. Your ovaries knew when to secrete estrogen, testes knew exactly when a little testosterone was needed. These glands were autonomous on their own. This turned out to be totally wrong, but that was the sense at the time, mainly because it was a lot easier to study what these glands did than study the pituitary. It just gave this erroneous sense that peripheral glands had their fingers on their own buttons, they could regulate themselves. And this gave rise to one of the truly silly incidences in the history of endocrinology, this craze that emerged around the turn of the century. There was this completely lunatic theory at the time that what aging is about, aging is due to testosterone levels declining, male sex hormones declining, and that's what causes aging. Testosterone levels go down with age, you lose your male vigor and energy, and you get old. You notice in the scenario about half the population is left out of the calculations there, but somehow, female aging was not a hot subject at the time. Aging, as in what's happening to males who are doing aging research, aging is due to declining testosterone levels. And this gave rise to this ludicrous medical fad at the time, which was to have people, aging males, injected with testes and testicular extracts from all sorts of unlikely laboratory animals. And this was this whole craze of very wealthy, moneyed gentlemen in their later years getting injected with monkey testes and rooster testes, captains of industries, heads of states, popes, things like that, all sorts of stuff. People going to these expensive sanitarium to get these very painful injections in their rear end, and what do you know? All of them reported these amazing effects. They felt better, they felt invigorated, all sorts of stuff. One big placebo effect, because it turned out those testes are not autonomous glands. They're not making up their own minds. Nothing's working at all. But that sort of placebo effect, if you're paying a fortune and you're getting these painful injections in your tush there, you've got a certain incentive to start feeling better. And these guys reported this, and it was great, and this was such a fad at the time, and this was nonsense. Nonsense built around this notion that the peripheral glands knew what they were doing. It took a couple of decades for people to figure out this was not the case. It was actually the pituitary gland that was telling these peripheral glands when to secrete their hormones. And take a peripheral gland and stick it in somebody's rear end or extracts of it, that wasn't going to do any good at all. The pituitary appeared to be running the show. And this gave rise to the archetypal description of the pituitary gland, and this is one that wound up in Reader's Digest. Remember that Reader's Digest series, I Am Joe's Pancreas, I Am Joe's Heart? Those series they're teaching you about physiology, where you'd sit and read it, I'm Joe's heart, I'm Joe's liver, I'm Joe's ovaries, that sort of stuff, where in the pituitary, 2 paragraphs into the article of Joe's pituitary, you would get that cliche, the pituitary is the master gland. The pituitary is the gland that's telling all these peripheral glands what to do. That's where the field was by the 1950s or so, and this was the gland that ran the whole show. Except people began to figure out there was a problem there, which was there were diseases that would pop up of people's hypothalamuses where the pituitary stopped making sense. or somebody could have some injury in that area in the base of the brain around the hypothalamus and the pituitary didn't do the right thing, or experimentally you would take an animal's pituitary, remove it, and put it in a petri dish, and by all logic, that pituitary, being this autonomous master gland, should continue to know what it's doing, and it would go crazy. Certain hormones it would stop secreting, other hormones it would secrete 1,000-fold more than usual. It began to dawn on people, and on one scientist in particular, that that pituitary was not a master gland. The brain was telling it what to do, the hypothalamus. And careful experiments at the time showed the hypothalamus was the master gland. And how could it do this? How was it doing this? Was it doing it with axonal projections? It turned out in some cases, axons going from the hypothalamus down to one part of the pituitary. In another area, the absolutely crazed theory popped up at the time that the brain was actually releasing hormones of its own. Hormones which got into this little private circulatory system between the hypothalamus and the pituitary, hormones that would go from the hypothalamus to the pituitary and stimulate the pituitary to release its hormones, which would cause the peripheral glands. People thought this was crazy. Number one, experimentally, how are you going to show this? This is a tiny circulatory system. Nobody's going to be able to show these imaginary brain hormones. The other thing was this bias at the time that hormones, yuck, maybe they come out of your kidneys or pancreas or whatever, but your brain isn't going to be secreting these disgusting hormones. What's it, some gland just dripping out these hormones? And that's exactly what it turned out to be. A pair of scientists did an extraordinary journey, spending the next decade searching for these imaginary hypothalamic hormones. A pair named Guillemot and Shalley, in a very tense competitive race of trying to be the first ones to isolate these hormones that half the other people didn't even think existed. Massive amounts of hypothalami taken from stockyards, massively inventive bits of biochemistry to try to isolate stuff, and it turned out this theory was right. Your hypothalamus did indeed contain hormones which got into this little circulatory system, told your pituitary what to do, which ran the peripheral glands. And Guillemot and Shalley got a well-deserved Nobel Prize for this, for showing the brain's a gland. The brain is an endocrine organ. The brain is the master gland running these neuroendocrine systems. And that's our modern view. Most of endocrinology is ultimately under the control of the brain in these systems. There's a few weirdo endocrine systems that seem to be independent of the brain. One example, fascinating hormone found a number of years ago, a hormone made in your heart. Look at this trend. First, your brain turns into an endocrine gland. Now your heart's an endocrine gland as well. A hormone there called ANF, and don't worry about what that stands for. ANF goes to your kidneys and promotes urine formation, promotes diuresis. What's this about? Your heart is asking your kidneys to make urine. Where does your kidney make urine from? It makes it from your circulation. It borrows water from your circulation. If you're making a lot of urine, you're decreasing blood volume. When does your heart want to ask your kidney to decrease blood volume? If you're suffering from high blood pressure. If you have a period of high blood pressure, your heart muscles are being extended or being stretched more as a result of the force. The stretching is the signal on heart muscle to secrete this ANF hormone, goes to the kidney, make more urine, decrease fluid volume, down goes blood pressure. Very clever trick. This is one system that's relatively independent of the brain. Most of endocrinology, though, is ultimately under control of that master gland, your hypothalamus. Okay. This allows us now to look at a little bit about what the actual hormones are, the structures of these compounds. And you remember back in that neurotransmitter lecture, lecture number 2, one of the things I really wanted to emphasize was one should not be overly impressed with neurotransmitters. One should not be intimidated by their chemistry. That principle of why you print newspaper on cheap paper, it's very simple principles. Make the stuff out of plentiful precursors. All of that cheapness of neurotransmitter chemistry, same business with hormones. Same exact need. These are just messengers. There's nothing intrinsically fancy about them. So looking at that, look how you'd make these principles work for you. Looking at a first class of hormones, and these are ones we've all heard of, these are steroid hormones. Steroid hormones are made of cholesterol. Cholesterol is the precursor molecule for all of these different steroid hormones. Cholesterol has this ring structure, and don't worry if this chemistry doesn't make sense to you. It's just a simple precursor, and this shows this theme. You use 1%, 1/10 of 1%, 100th of a percent of your dietary cholesterol to make all of your steroid hormones. What does that mean? Nobody on earth has ever run out of any of these hormones because they don't have enough cholesterol around. That usual principle where you want to have plentiful precursors, you don't want to accidentally run out of a hormone. So out of a cholesterol precursor, you can make as much as 5 different classes of steroid hormones. Estrogens, very relevant to female reproduction, androgens, like testosterone, progestins, like progesterone, and very important in female reproductive physiology, glucocorticoids like hydrocortisone, which were mentioned in the previous lecture, and a class of hormones called mineralocorticoids, one of which is called aldosterone, which has something to do with mineral regulation, water balance, electrolytes. The details of these are not important. Some of these we'll come back to with more information. The striking thing here is, look at all these different classes of hormones you're making out of one precursor. You're being cheap. You're being very efficient. This is that same principle over and over. Start with a simple precursor, one you're never ever going to run out of. Just with your cholesterol a little bit, and now you got a male sex hormone. And mess around with the bonds in another place there and put on a carbon here and a hydrogen there, and now you have a female sex hormone. You can be real cheap with the system generating extremely different messengers very simply out of a couple of precursors. Another example of the same principle is seen with a second class of hormones. And this second class is made out of not steroid molecules like cholesterol is, but these are made out of amino acids. These are made out of proteins. Proteins are sequences of amino acids. You take one amino acid and chain it to another one, another one, another one, and you make these long sequences, and these are called proteins. This is what a protein is, and there's many hormones that are made out of proteins. Insulin is a protein-derived hormone. Luteinizing hormone is another protein-derived hormone. A whole class of them, very straightforward chemistry. You're just taking these plentiful building blocks, these precursors, and just glue a string of them together, and this is insulin now. Here's one of the tricks that we've evolved for being very cheap about making these protein-derived hormones. And do not worry about the jargon here, but there's this one particular protein, a long protein, and schematically shown here, and this is not a hormone, this is a precursor to a hormone. And its abbreviation is POMC. I'm not even going to mention the real name because it's too horrifying to say on tape. It's this long, complicated term. It doesn't do anything. This is not a hormone. This is the precursor to it. This long protein contains a relevant hormone embedded in it. So what do? A very interesting trick. In your pituitary gland, cells in the pituitary synthesize this POMC precursor, and there's enzymes in the pituitary that snip here, and then other ones that snip here and here, and you've just generated 2 fragments. These are the hormones. These are 2 different kinds of hormones that are made out of this precursor. That's pretty efficient. You've just managed to make 2 different chemical messengers out of the same precursor. Same principle as use cholesterol as a precursor, and you could make lots of different types of steroid hormones. Here, use this big POMC thing as your precursor, and you cleave it in certain places, and you've generated 2 different chemical messengers. But look at the extra trick you do here. In another part of your body, in your adrenal gland, you splice this up differently. Here there's different enzymes, and they chop here and here and here and here and so on in these different splice places. And in the adrenal, you make 4 hormones there that are completely unrelated to these 2. You're cutting and splicing and editing and pasting together, and this is this remarkably efficient way to come up with a lot of chemical messengers from a very simple precursor. So that's a trick that you see with protein hormones. An additional trick, and a recently recognized one, is shown here. Again, don't pay too much attention to the jargon. That's not the main point. The point is how you paste these pieces together. What we have here are occasional protein hormones where they're made out of 2 different strands of protein. Rather than one single strand, you glue them together. And what we have here is a very efficient trick. We have this protein that has a gamma chain and a beta chain. Scientists love using gammas and betas all over the place. It makes us all feel more scientific. And what you see here is if you glue 1 gamma and 1 beta chain together, you create one kind of hormone. And if you chain 2 beta chains together, you produce a different kind. And the interesting thing, this is follicle-stimulating hormone inhibiting hormone, and this is follicle-stimulating hormone releasing hormone. This one stimulates something in your pituitary. This one inhibits something in your pituitary. They do totally opposite things. All you do is mix and match in different ways. Put two of these together, and you have the exact opposite effect than if you combine one and one. Again, this tremendous efficiency that you get there of just taking one precursor and generating multiple hormones, or just mixing different constituents, you get a lot of potential for efficiency there. There's an important implication of this, though, a very, very important implication, which is if you're being cheap in generating a whole lot of these chemical messengers that all look roughly the same, you haven't put much effort into making estrogen look all that different from testosterone, look all that different from glucocorticoids. If you're going to be cheap generating the messengers, you'd better have some very fancy receptors that can tell the difference. Because just imagine if you have organs in your body where they can't tell the difference between estrogen and testosterone. You've got some big problems at that point. If you're going to be cheap in generating the messengers, your receptors have to be very subtle and very expensive. If these two hormones, going back to this diagram, if these two hormones do exactly opposite things, you'd better have receptors that can tell the difference between them. You don't want a receptor that just keys in on the blue part at this end, because these two hormones will look exactly the same. It's got to be able to differentiate these two. So that's where you pay a price. As in much of life, in endocrinology, there is no free lunch. You get away cheap making your messengers, you got to spend a fortune on receptors that are very subtle and very discriminating, telling these little differences. So that's our second class of hormones. The first one, the steroid hormones. The second class, when you take these amino acids and put long chains of them together, forming these protein-derived hormones. The third class has a similar theme. There's a number of hormones where you make them from one single amino acid. And we saw that back in the neurochemistry lecture. Glutamate, we remember, is a single amino acid. Epinephrine, norepinephrine, those are made out of single amino acids with some modifications there. A bunch of the hormones are as well. Epinephrine serving as a hormone, as I just said, that's derived from a single amino acid. Some of your thyroid hormones are as well. Melatonin, a hormone that's very sort of contentious these days, whether it has anything to do with anti-aging properties. Melatonin is a hormone that's made of a single amino acid. Okay, we've got the building blocks now. There's nothing intrinsically impressive about steroidal hormones or protein hormones or single amino acid hormones. These are just the different ways of making these messengers. And we've seen something about how they go about their properties. Okay, so this shifts us to looking at the other half of it. We have the hormones, these are the messengers. These are the ones giving the signals, these are the ones shouting out that they have a message, and as we know already, the cell has to have a means to listen to it. The cell has to have receptors for them. Same principles that we saw with the neurotransmitters. And we see a version of that same principle yet again. We saw this before with the neurotransmitters. We have our standard picture up on top, a certain level of hormone, and a certain level of receptor at a target tissue. And the same story again that we saw previously, we can have down-regulation of receptors. If there's a massive increase in the amount of hormones, the target cells will tend to decrease their number of receptors. And conversely, we have our same phenomenon again of upregulation. If you have a decrease in the amount of hormone, the target cells will listen more clearly. Those same principles, you should be noting how similar hormones are to neurotransmitters, the same synthetic principles, the same regulatory issues there. They're just messengers. An additional feature that is shown by hormones is a very interesting one. There's a property you will often see. And actually, let's stick with this previous diagram. Can we put these up simultaneously? Okay, you notice this tendency, increase the amount of hormone, you tend to decrease the amount of receptor. Decrease the amount of hormone, and you tend to increase the amount of receptor. There's this counterbalancing, which is shown schematically here. There's an inverse relationship in a lot of these endocrine systems. You increase the amount of hormone, you're decreasing the amount of receptor. You would just say, well, what's the interest of this? Why have a regulatory system like that? This is presumably fancy stuff, up and down regulating your receptors. Doesn't it all just cancel out? If you double the amount of hormone and you cut the amount of receptor in half, or you cut the amount of hormone in half and you double the amount, it should all just balance out. Why all this fancy regulatory stuff where it sounds like it just cancels everything out? Turns out it doesn't cancel things out. It does something much more subtle. The ratios of these up and down regulatory changes, there's not a one-for-one correspondence. The typical thing that you see is when you combine an increase in the amount of hormone and a decrease in the amount of receptor, when you put the pieces together, you tend to get a pattern that looks like this. You get an inverse U pattern. And we've already seen an example of that, a hormone working that way. You remember glucocorticoids and their effect on LTP, long-term potentiation. Go from no stress to moderate stress, and you enhance memory formation. Go from moderate stress to lots of stress, and things get bad again. It is a very rare trait in any sort of physiological system that a moderate amount of something is moderately better and a whole lot is a whole lot better and an infinite amount is infinitely better, that it just keeps going up or just keeps going down. Instead, lots of physiological systems show this property. A moderate increase in some pattern, a whole lot of it you go the other way. Inverse U's a point of homeostatic balance, and this is how you construct it. You construct it by having opposite effects of the messenger and the system that responds to it. Put those pieces together, and in a lot of physiological systems, you get this endpoint. Very, very interesting. Okay, now what we've managed to do now is get a brief overview of the endocrine system. And there's a certain simplification I've been putting through here, which is the hypothalamus has one class of hormones which tell the pituitary to release one class of hormones which influence events in your kidney. Meanwhile, over on the next farm, the hypothalamus has another class of hormones that regulate different hormones in the pituitary that have something to do with your thyroid gland. And then over in another part of the hypothalamus that there's compartmentalization among these hormones. In terms of what organ they affect, in terms of what physiological effects they have, this is how people typically get taught endocrinology because it's complicated enough as it is. There's a certain pressure to group things in terms of these endocrine systems. The most modern picture of all of this instead is you're getting a tremendous amount of regulation between these different endocrine systems. And this is dizzyingly complex, and I'm going to show one example of this now, an extremely interesting one, and one that's rather relevant. And this is the converse to the story shown in the last lecture showing why it is that when you're under stress, penises don't work very well, and erections and all sorts of problems with that. Stress and its effects on male reproduction. What we'll see now is stress and its effects on female reproduction, because this is a classic example of this crosstalk between different endocrine systems. Okay, we will be coming back to this diagram over and over again here during this stretch, and what we see here is the basic workings of the female reproductive system. And we'll work our way down here, beginning with the brain, pituitary, and ovaries. And we see some of the relevant hormones here. Coming out of the brain is a hormone called LHRH. Do not worry about the jargon here. This is not critical for understanding this. LHRH, luteinizing hormone, releasing hormone, those of you who want to get the jargon down. LHRH, this LH releasing hormone, causes the pituitary to release LH, luteinizing hormone. Along with that, out comes FSH, follicle-stimulating hormone. These are terms many of you should be familiar with, and collectively what they do is cause the ovary to secrete estrogen and progesterone and cause egg maturation. Progesterone, in turn, works on the uterus to thicken the uterine wall. This is the basic endocrine system of females. Here we have an additional very interesting step which becomes quite relevant to understanding stress. What we have is this odd trait that pops up in females. Females the world over, humans and non-humans, secrete a certain amount of androgens. Androgens, male sex hormones, derived from testosterone, close relatives of testosterone. What are females doing secreting male sex hormones? Nobody has a clue. Another one of those mysteries of nature. Nevertheless, androgens are secreted by females. They come from the adrenal glands of females. These are called, thus, adrenal androgens. And it's not a ton of the stuff, maybe 5% the levels you would see in a male. Nevertheless, you got to do something about them. You can't have these sitting around. They will cause problems. You've got to get rid of them. And you see a very clever trick that females have evolved, making use of fat cells. Fat cells contain an enzyme which converts androgens to estrogens. This is simple stuff. We know this from a couple of figures ago. These are both steroid hormones. They're both derived from cholesterol. They're very close to each other structurally. And biochemistry 101, Fats learned how to do this long ago. Very simple conversion. Androgens get converted to estrogens. You're home free. Everything's taken care of. Simple intervention. Great. What if, however, you don't have a whole lot of fat around? What if you're starving? What if the locusts have come and eaten your crops, and for the next 6 months, you're foraging 20 miles a day to get enough food, and your fat deposits are going down? If you don't have enough fat, you're not going to have enough of that enzyme around. And suddenly, you're not converting androgens to estrogens anymore. And you get into trouble at that point. A minor reason why you get into trouble is there's less estrogen in your circulation. The bigger reason is these androgens are building up and accumulating, and they are going to wreak havoc at the level of the hypothalamus and the pituitary and the ovaries and the uterine wall, and all sorts of stuff doesn't work well. That's the mechanism by which starvation shuts down ovulation in females. And that's real relevant for understanding animal populations out in the wild and how populations are regulated during periods of starvation, famine, things of that sort. That's also relevant to humans and a certain subset of humans in our society. That's why you stop ovulating if you were voluntarily starving. Anorexia. People with anorexia, predominantly girls and women who have anorexia, a hallmark of the disease, as you lose weight, you stop ovulating. And this is one of the mechanisms for it. Very interestingly, this is also relevant to understanding why women who do lots and lots of athletics often begin to have problems with their cycles, begin to be anovulatory, amenorrheic. With enough exercise, your fat content drops down enough, and there's a critical muscle-fat ratio, after which you stop ovulating. And all sorts of studies have shown this. Girls who do lots and lots of exercise, typically runners, ballet dancers, things of that sort, gymnasts, they have a significant delay in the onset of puberty. And women who do lots of exercise, they will often get below a critical muscle-fat ratio, and they will stop ovulating. Obvious sort of question here is how much of it do you have to do to get into trouble? You know, it's a lot easier to quantify how many miles you run per week rather than how many pliés in a ballet class. The general number that comes out of the running studies is if a woman is running 45, 50 miles a week, that's typically the point at which you begin to see some problems. And this is a very important lesson for us. This is a very important lesson, and this applies just as much to male reproductive physiology. Men who are running 45, 50 miles a week are often beginning to get reproductive problems as well, other medical problems. In either sex, that much running is associated with some osteoporosis of bones, some metabolic problems that have some resemblance to diabetes. Not in everyone, but you begin to see the first hints of this. This teaches us something very important. Somebody sits you down and informs you that exercise is a good thing, and yes, indeed, exercise does turn out to be a good thing. It is much better to exercise sometime rather than be a couch potato. That helps your body function better. But it is not the case that just because exercise is a good thing and a fair amount of exercise is even better for you, that massive amounts of exercise are going to be massively great for you. It's an inverse U. There's very few things in physiology where the more you do, the better it gets, and all you have to do to get a sense of this in terms of exercise is, here's what you do. Sit down, in your mind, sit down some hunter-gatherer from the desert in Central Africa, and sit him down there and say, you know, where I come from, we've got so much food and so much free time that sometimes we just go out and run 26 miles for the sheer pleasure of it. And they're going to sit there and say, are you crazy? That's stressful. This is really bizarre behavior from a hominid standpoint. In general, throughout hominid history, if you're running 26 miles in a day, you are either very intent on eating somebody or somebody's very intent on eating you. This is not normal hominid behavior. When you look at people out in non-westernized settings, this sort of stuff is not what our ancestors spent all their time doing. And if you do this on a regular basis, this is one of the ways in which you get into trouble. A very important take-home lesson in physiology: everything has its point of balance of homeostatic optimum. Okay, so that's one way in which you can get into trouble if you are female and under stress: physical stress, starvation. But what about stress, the sort of everyday psychological stress that plagues lots of us, problems there? And looking at this entire axis here, this entire cascade, what you see is the potential for psychological stress to wreak havoc. If you were psychologically stressed, you've got a variety of hormones that you are secreting, putting into your circulation. One called beta endorphin. We've already heard about glucocorticoids, another hormone called prolactin, and these work at every step along the way. Beta endorphin and prolactin working at the brain to block LHRH release. Prolactin and glucocorticoids working at the pituitary to block the sensitivity of the pituitary to LHRH. Glucocorticoids working at the level of the ovaries to block sensitivity to LH. So look at this triple whammy. You're making less of this hormone. The pituitary isn't listening to it, so even less of this is coming out. The ovaries aren't listening there. Just a smidgen of estrogen and progesterone are going to come out. That's hardly any progesterone to work on the uterine wall and cause it to thicken. And on top of it, prolactin blocks the effects of progesterone on the uterine wall. So if you've managed to actually somehow inconceivably still have enough of these hormones dribble out of the ovaries and manage to get an egg fertilized, you're not even going to implant it. Everything stops working up and down the system, and these are the things going on during periods of psychological stress. This is not starvation. This is not lions running after you. This is psychological factors. In terms of those hormones that regulate that system, and what's striking here is that these are hormones from other parts of endocrine systems working on this endocrine branch. This is this crosstalk I told you about. You've got the beta endorphin, the glucocorticoids, the prolactin impinging on the system. Quantitatively, probably the most powerful of those hormones is prolactin. Prolactin is real strong in its anti-reproductive effects in females. If you don't feel like ovulating, prolactin is the hormone that'll do it for you. Prolactin plays a central role in psychological stress suppressing reproduction, all sorts of other stressors as well. Prolactin has something to do with nursing as well. Very interesting feature here. When you nurse, when one nurses, one secretes a ton of prolactin out of the pituitary. There's a reflex loop going from the nipples in both sexes up the spinal cord into the autonomic nervous system, triggering the hypothalamus to tell the pituitary to do something, and out comes prolactin. Nursing stimulates prolactin release. Prolactin suppresses reproductive function, as you just saw. That's the reason why nursing is such an effective form of contraception. And I know all of you are out there saying, oh, that male, what does he know about it? Nursing doesn't work all that well. Nursing works spectacularly. The World Health Organization estimates more pregnancies are prevented each year with nursing than every other form of contraception put together. All you have to do is nurse properly, which is what I'm going to teach you to do now in the privacy of your own home. Okay, here's what this is all about. It's got something to do with the pattern with which people nurse. Women in Western societies, how do they nurse? They give birth, they pick up the kid, and they nurse once every 3 hours or so for half an hour at a time. After 6 months, if they're still nursing, they'll nurse every 6 hours or so for half an hour. What you see then is you begin nursing, up go prolactin levels, and you stop nursing, and an hour later or so, it's back down, and it scallops up and down like this. This is not how women usually nurse. In most of the world, this is not how our ancestors nursed. You get a very different pattern. And this pattern was discovered a few years ago by some anthropologists looking at these hunter-gatherers, these Kalahari Bushmen. These are the folks in the movie The Gods Must Be Crazy. These are folks wandering around the Kalahari Desert in Botswana, and anthropologists have studied these folks for years, and they've got something very strange about their reproductive lives, which is these Bushmen men and these Bushman women have sex all the time. They have no ritualistic prohibitions. They have no contraceptives whatsoever, yet women give birth once every 4 or 5 years. Why aren't they getting pregnant in between? Well, everybody used to have a very easy explanation for this, which is they're hunter-gatherers, they're obviously starving, they're short, nasty, and brutish, they're having a tough life, they're all fat-depleted, and obviously the women aren't ovulating. People went and looked at them closely, anthropologists in the 60s, and discovered these people were not starving. These people were having a very comfortable existence in lots of ways. If you're going to be in the developing world, you would much rather be a hunter-gatherer than an agriculturalist or a nomadic pastoralist. You have a much better diet, far more variety, far more resistance to famine, because you're eating 300 different plant species instead of the 6 or 7 things you're cultivating. You have far less work. Agriculturalists work like maniacs. These hunter-gatherers work 3-4 hours a day for their calories, and they spend the rest of the time doing social anthropology or whatever they do there. It's a much easier lifestyle. Agriculture was one of the great stupid mistakes in human history. That was a bad move. The main advantage of agriculture is it allows you to concentrate capital and resource in the hands of a few people and start making stratified societies and all sorts of nasty stuff. Hidden political agenda here? I don't know. But what you wind up seeing there is hunter-gatherers are not starving. The term given for them is this was the original affluent society. These people were doing just fine. So there goes the, oh, they're all depleted of fat argument. Nobody knew what was going on until an anthropologist physician named Mel Connor went and studied these folks, and it turned out it's the nursing. These women nurse very differently than women in Western societies. What do there? They give birth to a kid, and they pick up the kid, and they nurse the kid for 30 seconds. And they nurse the kid every half hour or so for 30 seconds, around the clock, for the next 4 years. Aha, that doesn't sound like such a great deal after all. Maybe you're not going to try that. Totally different pattern. Nursing all the time, little short bursts of nursing. They carry these kids around on hip slings where the kids nurse every half hour at night. The child is sleeping with the mother and the father, and the parents don't even wake up. The kid wakes up and nurses there. Everybody else is sleeping perfectly comfortably, except for Connor, the anthropologist, there with his night viewing goggles watching these folks in his little stopwatch, but everybody else is perfectly comfortable. 4 years old, the kid's still nursing, they're out hunter-gathering, and the kid's off at a Little League game, and every half hour runs back and nurses for 30 seconds. What you see then is a very different pattern, which is you nurse the first time, up go prolactin levels, and they're staying up for the next 4 years or so, and you're not going to ovulate no-how then. When that's going on, you have a very different pattern, and the sense is that's the typical pattern throughout human history. That's the pattern in non-human primates, very different things going on. Think about the implications of this for a second, something that probably half of you can appreciate far more than me. Think about the life histories of these women. They reach puberty when they're 14 or so, a little bit later than in the West. Have a few cycles, get pregnant, give birth to their first child, wean the child when they're 19, when the mother is 19 or 20, have a few cycles, get pregnant again, wean that child when they're age 25, a few more cycles. They reach menopause around age 40, earlier than the West. Think about the implications. These women are having 9-10 periods their entire life versus 300 or 400 amongst Western women. Completely different pattern of physiology. And a number of people have speculated that various diseases, obstetric and gynecological diseases of women in the West, are diseases that are due to taking this big, massive, complicated, expensive piece of physiology that evolved to be used a dozen times and running it 300 or 400 times. And when you run it 300 or 400 times, a lot more glitches pop up. The best example of that is endometriosis. Endometriosis appears to be a disease that you go for too long not getting pregnant. You increase the likelihood of endometriosis. Then this appears to be a disease of the thickening and the sloughing of the uterine wall over and over and over and over and over, when it was meant to only happen a dozen times or so. A disease of the West. We have a fair number of diseases of the West. We have invented a very, very atypical lifestyle for ourselves, very different from our ancestors and very different from how most other primates are living out there. And when you do that, you got some pluses, but you got some minuses. This appears to be one of the minuses. Okay, so what this lecture has brought us to here is seeing the basics of the endocrine system. How you regulate endocrinology, and much of it at the level of the hypothalamus, your brain once again having enormous control over what's going on, your different classes of hormones, the main point there being you shouldn't be overly impressed with any of this. They're just messengers. Finally, we've seen how receptors are important in the story as well. You can't make a message without somebody being there to listen to it, and how you regulate sensitivity at those two ends, and putting the pieces together just now, looking at this very complex endocrine system. What we need to do in the next lecture is tie it into the theme in this course. We now know the basics of endocrinology. What does this have to do with behavior? How do hormones affect our brain, affect our behaviors, our emotions, and as always our theme of the individual differences. Okay, so that finishes up here. Once again, time's almost up and it's time for my nap. So why don't we stop now and we'll resume again on the next lecture tomorrow.